Thank you. And first of all, I'd like to note that I'm talking to you from Naarm, Melbourne, Australia, on the lands of the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders, past, present and emerging, and any elders from Australia who might be on this symposium panel tonight, or not on the call tonight. So we have a pretty big panel lined up. The speakers have been allotted 20 minutes, with, I understand, about 15 minutes for their presentation. Potentially a question after their presentation, but most of the discussion will occur when we get to the panel session. And I have decided, because I can, that we will have a 10-minute break after the speakers, because I think it's cruel and unnecessary treatment to have people sit for 3 hours without having a chance to stand up. So I understand, Dave, that you have to leave and won't be around for the panel discussion. So we will take questions for Dave straight after his presentation. I am, as Stuart knows, fearsome about timekeeping, so I will let people know when they have 4 minutes left. Yes, and unfortunately, I don't have control of switching you off. But 4 minutes left, and then 1 minute. And if you could wrap up, then it just makes sure that everybody gets their opportunity to speak. So, firstly, it's a great pleasure for me to introduce Dave Stewart, who's the director of Instruct-ERIC, which I understand is involved in structural biology, shared infrastructure, and also director of Diamond, which is a synchrotron located at the University of Oxford. So over to you.
Hi, thanks very much. Shall I share my screen and get going?
Yes. Yes.
I think that's a good idea, isn't it? Okay. Can you see my screen and hear me?
Yes.
Okay, great. Yeah, well, thanks very much for the introduction, and apologies I have to leave, but we have an Instruct Council meeting today, so it's a big day for us. So yeah, so I'll be talking on behalf of Instruct, but as you say, I've also got a position as Life Science Director at the UK synchrotron near Oxford, and I'll highlight some of the stuff that's been going on there. And just to briefly introduce Instruct, it's a European research infrastructure. It's distributed. The idea is to work with structural biology researchers, but also to try and open up the field to more broadly. And the whole sort of nature of this obviously is collaboration and shared experience. Instruct was set up in 2017, but actually had as an ERIC, but has been operating in a fashion since earlier. So it's been going for some time, providing access to structural biology instruments and resources, and also doing things like training. So for people that are not structural biologists, the idea of structural biology is essentially to link the molecular and atomic level to the cellular level to try and explain biology in sufficient detail that you can make chemical interventions and understand mechanism. So it's basic science, most of it, but it's also heavily translational. So most of the use of facilities like synchrotrons by industry is by the pharmaceutical and biotech sector. So it's recognized as crucial to drug discovery. So the scientific impact of Instruct is partly excellent science. So Instruct was involved in 226 publications in 2020, but also the idea is to keep structural biology is dependent on technical innovation. So the idea is that to try and keep that technical innovation going and develop new methods, and not just develop them, but make them available to users to democratize cutting edge methods. And as I've said, structural biology also has a significant broader impact, not just in drug design, but vaccine design, diagnostics, and of course the usual career development, etc. So, although we're a European research infrastructure, we are interested in making global links, and we've been mainly working with South America and Africa, some links with India and China. But at the moment, although Europe is well organized to set up pan-European research infrastructures, that I don't think we've really got the clear mechanisms to make these things global. So we've been really hindered there. I will just now give a couple of scientific examples to give an idea of what all this actually means. And the first is really they're focusing on responses to the COVID-19 crisis. The first of those is using crystallography to determine structures at the diamond synchrotron. And this work involves very many groups from around the world. And here there's a map of the viral genome, and the take-home message is that a whole host of potential drug targets have been had their structures determined at Diamond by, as I say, by a variety of groups. So there's key antiviral targets have been identified and taken to some state of readiness for drug discovery. And I'll just focus on one of those, which is the most mature of the targets, which is a protease. So this is an essential protein that chops up the viral polyprotein as it's made in the cell and it's required for the virus to be active. And this started with a phone call from Mayrhofer, a long-term collaborator of ours who works in Shanghai in China. They had some issues because their synchrotron was shut down for a while, so we initiated a collaboration with them. That was the end of January 2020, and very quickly, with their help, it was possible to get structures. And then at that point, by mid-March, through a collaboration fostered by Instruct involving particularly chemists in the Weizmann Institute in Israel who do covalent chemistry for covalent inhibitors, it was possible to determine the structures of a large number of small chemical fragments bound to this drug target, and they were released immediately on the web. And there was then some Twitter sort of storm that got the attention of people for this, and that rapidly led to setting up a rather informal but global collaboration to take these forward to the next stage. This involved academics, people, a lot of it people from industry who were sort of not doing very much at that time because there was a lot of lockdowns, and also some industries that gave pro bono chemical compounds to this. In brief, that rapidly developed so that, well, it developed rapidly, and then there were a few bottlenecks in terms of getting assays, getting animal experiments done. But now the state of that is that it's there's been substantial funding announced recently, which will take it through in the next few months to see if it's ready to go into the clinical trials development. So preclinical research is underway. So that's an example of an unusual sort of forms of collaborative research driven initially by social media. We've also done more, there's also more standard collaborations that have gone on with industries involving massive screens to try and repurpose drugs. And this involves a number of large companies who are expert in artificial intelligence trying to bring AI into this field in a more effective way to accelerate drug discovery. There's other techniques in structural biology that have also been relevant during the pandemic. Electron microscopy is now a way of getting atomic level detail on very difficult targets. And this has been really very important in understanding how the virus gets into cells, assembly and production of the viruses, but also how antibodies neutralize the virus and how the virus escapes that neutralization. So I haven't got time to go through the details, but there's a whole series of papers that have emerged on that basic sort of academic research. In 2021, there was a series of 5 Cell papers, for instance. To just broaden it out now, to give 2 specific collaborative examples of work. One of those is in the area of COVID-19. So this is to show how you can have collaborations outside of structural biology that can be useful in accelerating things. And this is a team from Janssen Vaccines who worked with Instruct in the Netherlands to help understand, develop, and characterize new stable variants of the spike protein. And essentially, they're using structural biology to understand what's going on here. And you should be aware that the majority of the advanced vaccines that are now in use not the sort of wild type spike, but an engineered spike, which is based on the structural biology understanding that has allowed us to improve the physicochemical properties and gives a more potent antigen for these vaccine developments. So that's one, and that I would say this is really the tip of the iceberg, and the take-home message is that structural biology is a very powerful tool in this area. And openness, sharing, collaboration, open data are really critical to making the most of this. Another example, completely different now, is understanding how nerves function. There are these molecules called GABA receptors, which provide channels which chloride ions can go through in the central nervous system. And these are targets of a number of mental health drugs. And here there was a collaboration between LMB Cambridge, where they were working on the structure of this, but they were struggling to identify the complex structure. So there was a collaboration established with the center in Belgium, where Instruct Center, where they developed some technology for developing specific binders that were large enough to act as markers to allow structures to be determined. So this really opened the door to doing high resolution structures of these important molecules, and in turn now in subsequent papers has allowed a whole series of potential drugs to be visualized bound to this molecule. So that's just a couple of quick examples. And I should just finish by saying that these nanobodies, as they're called, these specific binders, have a whole range of general sort of values. So about just under 2 weeks ago, there was another paper published using nanobodies, in this case, as potential therapies against SARS-CoV-2. They're small enough that they can be potentially inhaled and act directly in the upper airways. So I'll stop at that point. I hope I haven't gone too much over. Thanks very much for your patience.
Thank you very much, Dave. That was a really terrific presentation and some great examples of the Instruct collaboration. I'm just quickly checking. I can't see any questions at the moment, but well, there are 2 things that particularly interest me. One is around the role of social media, and I was very interested that that very large collaboration took off because of social media. How did that sort of work, and do you think it's something we should be using more of in the future?
Yeah, I'm not an expert on social media, but this was Twitter, and it was started because somebody that works at Diamond, my deputy Martin Walsh, had an existing Twitter account and was followed by a number of people. So as soon as results started to come through, he sort of socialized this, and that was picked up and spread like wildfire. So yeah, I mean, it is something that I think Instruct does pretty well actually, but it's certainly worth thinking about how we make the most of this. It can be extraordinarily effective. Yeah.
Yeah, especially I guess across the world where time and things are so different. And I'm thinking of that obviously here in Australia where the sun is setting now. And you know, we have a few time zones, but not, the time is not nearly as different as between us and you. And how do you think Australia might become involved in this? Because I noticed on your maps that you didn't quite reach us.
I don't know. I think there's real opportunities. There are that. I know that in Diamond we already have in some areas collaborations with the Australian synchrotron, and those are very successful. I think there are real and there's some very good work going on in the sort of drug discovery area in Australia that would be sensible to link up with. It's been a bit haphazard. I think if there are ways of looking a bit more systematically for doing it, but as you say, the time difference is a bit of a killer, isn't it?
Yeah, it can make things pretty difficult for us here in Australia, but that happens in relation to North America as well. So so I guess there's a lot of excellent structural biology that is done here in Australia. So and I presume, I haven't had a look yet, but I presume your website would have details that people could connect up to explore opportunities. And
certainly, you know, Susan is on the call, and you know, she's very happy to directly sort of talk to people, as am I. So you know, just get directly in touch. Anybody that's got ideas, that'd be fantastic.
Okay, terrific. Susan, did you want to add anything to that?
No, I think just to emphasize what Dave said, that we're very interested to explore opportunities with groups in Australia and national research infrastructures in Australia as well. I think there are a lot of opportunities for working together. Now, especially when a lot of our infrastructures are available for remote access, or even just digital exchange, then that sort of negates some of the problems associated with time differences. So I think it's definitely worth conversation. We'd be very interested in talking to people about those.
I don't know if we have any Australian structural biologists on this call tonight, possibly not, but anyway, we might take that back to the community and see what we can do. If there's anybody who'd like to say anything, I can't see all my hands at once, so I can't see a hand there. Okay, so we might move on now. Thank you very much today. That was terrific, and good luck with your subsequent meeting.
Thanks.
Okay. Thank you. And we'll move on to the next presentation by Stuart Newman, who's the CEO of Therapeutic Innovation Australia. Off you go.
Thank you, Stella. I'll just navigate the sharing of the screen. How is that? Is that looking good? I'm not on the dreaded presenter view. Terrific. Okay, no, well, thank you, everybody. Okay, thank you, everybody. Stella, I'll do my best to keep the time because I know how you can be. So I'm going to talk a little bit about collaboration between complementary but quite different research infrastructures within Australia. But I think first, before I'll do that, I'll have my own little time zone narrative. TIA is part of a global group of similar organizations called Translation Together, with groups from the US NIH, from ELIXIR in the EU, and Japan, and most recently Brazil. And we have meeting every month or so to discuss initiatives, and the meetings flip between 2 times, either 6 a.m. in the morning or 10 p.m. at night for Australia. So whichever way we go, rather than me getting up early or staying up late. In fact, I have a meeting with them later on this evening to give another presentation. So we try and make it work how we can. But as I noted earlier, I'd like to talk. We've heard a lot about the NCRIS from Bernadette yesterday as well, so I'm not going to dwell on that too much, except to go straight to the end in big friendly red letters. And the C is NCRIS is most definitely collaborative. And so noted, we've heard a lot thus far on Australian facilities and projects working effectively and for some period of time with the equivalents in Europe. But I'd like to focus a little bit on, as I noted, how research facilities within Australia have started to work together or work more closely together and formalize their methods of working together. So in that respect, what I'm going to do is talk about these a group of NCRIS facilities that are starting to talk a bit more frequently and establish some joint initiatives together. To do that, I'll talk about introduce each of these facilities briefly, and then come back to the end with some examples of some projects and initiatives, and hopefully, I think, spark a bit of discussion and some questions later on. So what I have here on the left is a notional articulation of a typical innovation life cycle. This particular cycle describes a product or a therapeutic product or a diagnostic, but it could easily be adapted to a digital or public health intervention, starting with population health, looking at examination of disease burden and unmet need, into looking at the disease itself, its pathology and biology, examining how we might interact or modulate a disease state by throwing a target, then developing a way to either identify disease or a potential treatment, then through the drug discovery, drug development cycle, through the trials, and then feeding back finally into patient outcomes, which goes back to population health. So it's a bit of a cycle, and it's one way in which we can articulate the research or health innovation journey. And a feature of this cycle is that researchers or indeed companies can access facilities or capabilities anywhere in this cycle. And in this particular concept, each of these individual facilities can contribute to different parts of the cycle. I don't want to sort of imply that this is a totally sequential pipeline. So I just want to start off briefly with population health in bold here. So I'm just going to do my best to talk about some other of our NCRIS projects. So the Population Health Research Network is essentially about data, linked data from Australia's various states and territories and across the Commonwealth system in describing education and birth deaths and prescriptions, hospital records, all manner of data drawn from public health jurisdictions across the whole country, brought together in a very nicely managed and federated system with access available for researchers. I'll go to the some of the numbers first. There's a vast array of data and collections and a vast number of records. And this can be accessed in a manner of ways to inform research projects. One example, for example, is cervical cancer vaccine, and access to this data changed the methodology of cancer screening. And likewise, data has been used to inform clinical trials. BioPlatforms Australia, of which the BioCommons is a key part, which we just heard about, is a broad conglomeration of capabilities and capacities in the area of broadly in omics and bioinformatics. Genomics, proteomics, and metabolomics capability is accessible and kept up to date and is accessible across the country. I think the BioCommons has just been noted very recently. And quickly, here is a bit of a very impressive articulation of some of the national data projects which are responding to specific missions or challenges, whether it's a koala genome or Great Barrier Reef sequence, or indeed Chardonnay sequencing, and I'd love to go to that particular conference. The point being that this data is available and is reused to contribute to these missions of national and international importance. Genomics Australia, as we heard briefly about from Steve's and remarks earlier on, provides infrastructure in discovery and molecular base of disease, provides infrastructure to support the development of genomic-based medicine and precision medicine, to enable diagnosis, understanding, and treatment. There's 9 facilities around the country, and they support about 400 projects a year. And this is a bit of an articulation of where in the genomic, the phenomic cycle, Genomics Australia deals in. These orange circles show where phenomics has some capacity in. Again, there are some nice case studies for all of the facilities I'm talking about, including COVID antiviral therapy and some diagnosis in lupus. And I think Steve noted this slide earlier on. I'll talk a little bit about TIA. We park ourselves right in the so-called Valley of Death. And we express where we are in terms of technology readiness levels because we are a product development consortium. We've organized ourselves in 3 product-focused capabilities, namely small molecules, pharmaceuticals, biologics, and in the cell and gene therapies, so very much an emerging area. We've chosen about 24 national facilities across the country. And like the other facilities that I'm talking about, we provide access for research and SMEs, very bespoke services. And interestingly, each of these capabilities is supported by a project chaperone, a person who's dedicated to broker access to these services for industries and researchers. So I'll just very briefly go through each of their capabilities because they each have their quirks. Our small molecule pharmaceutical capability is very much drug discovery and development, starting with access to a national compound library. And I know the Compounds Australia is working quite closely with Bana and his colleagues at EU OpenScreen, and we're keen to see how that develops in the future. We support high throughput screening and medicinal chemistry and DMP guided optimization through to preclinical accredited preclinical efficacy studies. We used to have GLP toxicology in the field, but we no longer fund that because it was essentially privatized. Our cell and gene therapy capabilities spread across the country in a number of medical research institutes and hospitals. We essentially fund GMP accredited manufacturing of cell and gene therapies for clinical trials and direct patient use. and they provide advice in the regulatory environment, manufacturing and process development in things like CAR-T, gene therapies specifically, and immunotherapies particularly, and over in the West, tissue regeneration therapies as well. And a new addition to this capability is vector and genome engineering at Westmead, which is a very exciting new development. Our national biologics facilities, 3 nodes in Brisbane, Sydney, and Melbourne, essentially around the discovery and development and small-scale manufacture of biologics and vaccines. 2 of these 3 nodes were deeply involved in the development of a homegrown vaccine using a molecular clamp technology, which unfortunately the trial was halted a little bit prematurely, but it did go to show that we do have the capability onshore to be able to do some develop a vaccine, take it all the way to phase 1 and potentially beyond. We have just expanded our capability to include mRNA production at a lab scale and lab quality in response to demand for this new therapeutic modality. That's the last slide on TIA. In any case, is we run a voucher scheme, and we've found this to be a very useful mechanism for enabling access to our facilities. It's essentially an additional little subsidy that is assessed internally with some external advice. It's open to SMEs and academics, and also to international partners as well. I'd like to make that clear if there's any potential access users. We do this every 6 months. We've got a round open at the moment, actually. The advantage of this is a very rapid turnaround scheme. It enables some very quick killer experiments, which you need at this stage of drug development. We have a couple of case studies, and happy to talk about this a bit later on. I've presented this to other European colleagues in the consortium that I mentioned earlier on. The final group I'd just like to briefly talk about is National Imaging Facility, providing a truly world-class expertise in a number of modalities around the country, from PET and CT and MRI. Some big numbers involved, 55 open access instruments, 13 sites around the country. In the health space, a great deal of their activities in the health space, but they also across other areas as well, in environment, materials engineering, and even anthropology. Again, they are spread very much across the country in a number of jurisdictions, but all very well networked and highly accessible. And just a few areas in which they are looking to expand in the future, really beefing up the various technological platforms that they specialize in. So I think I have about 5 minutes. So I'll briefly run through these particular facilities. I just want to come back to this cycle diagram. And this diagram on the left is something that we're starting to develop. It's not particularly finished yet, but it's a way of articulating how we are already actively working together as groups, and how we are contributing to the same projects and the same initiatives. For example, Phenomics and bioplatforms are seeking to jointly appoint a sort of project facilitator for a specific piece of work, and for a half of a person, and the other half TIA is looking to employ as well. So in that way, we have a person who will be a real nice bit of connected glue between those 3 facilities. And again, there's various other schemes that we collaborate with. One that's mentioned a few times here is the MRFF, which is the Medical Research Future Fund, which is a large-scale Australian funding scheme, which is very mission-based. And we are, with the leadership of BioPlatforms Australia, engaging with this particular mission, which is related to cardiovascular health, as a group, in a way that we can articulate in one way, in one go, rather, what capabilities we can offer and what capabilities are accessible to researchers who are part of that particular mission. And something that Steve noted earlier on is the infrastructures are people, and it's the people that form relationships, and relationships is where the value is, and where the value is added, where the information is shared, and where the initiatives are formed. And this is some real benefits, I think, us working together, besides from the specific initiatives that mentioned earlier on. We are regularly sharing intelligence to
4
reduce
minutes, Stuart.
Thank you, Stella. Reduce duplication of effort and investment. We have monthly meetings and quarterly meetings of our overseeing chairs. They can contribute to the strategic discussion. We maintain knowledge of what each other do and what we can do. So it puts us in a position to refer users that come to us to other projects that may be able to assist them. And we do that quite a bit, actually. And as I noted before, the joint appointments that we're plan to put in place will facilitate this cross-fertilization even further. I think what we are starting to see as well is we're able to apply our specific expertise to individual projects at different stages of their development. For example, there are some projects that BioPlatforms has supported through access to their capabilities that we have gone on to support at a later stage in its development through some access to specific production and bioanalytics. So we're starting to see application of these capabilities for the same project at different points in the project's life cycle, which goes back to that cycle diagram. And I think I'm just going to stop there, Stella, and come in under time. And just say thank you very much for this opportunity. I'm pleased to elaborate on any of these points. I know that most, if not all, of the directors of the other 4 NCRIS projects are online to answer the questions that I was not able to. Thank you very much.
Thanks, Stuart. Natalie, did you, we got any, we have got a hand up there. Would you like to ask a question now? We have a minute before the next presentation. You unmute yourself.
Yes, hi. Thank you, Stuart, for this excellent talk. You briefly mentioned that you have an interesting voucher system to subsidize projects, but not to fully fund research projects. So in order to have this buy-in from the applicants, but basically means that the applicants, they need to be successful in getting a research grant in the first place, and at the same time that they need to successfully apply for the voucher. Could you tell us a little bit more about your experience so far with this subsidy, this voucher system, how well it works?
So yes, people do need to be able to bring some funding. There's a phrase that I hate, but it's very apt in this case. It's skin in the game. It's people have to sort of be committed enough and have validated the project enough to have found some funding, whether it's through discretionary funding from a university or a slush fund of a researcher, or a grant, or from a technology transfer office, or whatever. It's also important that the project is kind of pre-validated by the fact that it has been awarded some funding by somebody. That gives us confidence that it has already been through some form of peer review in a way, which gives us the confidence to know that they're serious as well. So that works particularly well. The other part in terms of having to apply for the, sorry, yep.
Sorry. I'm going to stop you there, and we might have a bit of a broader discussion about the voucher system during the panel discussion, if that's all right, just so we can move. So you can run through it in a bit more detail. I guess one thing to say is that the amount of the vouchers is not huge, maximum 50,000 Australian dollars. So people don't have to have huge amounts of money. Just quickly adding that in.
Yes, and it's also for very specific parts of a project, not for a whole project.
Yeah, and I think it'll be great, and Stuart will have more opportunity to describe that a bit later, because it's a very successful scheme that's been taken up by a number of other entities across Australia, a voucher scheme system. Okay, so our next speaker is Michaela Mayrhofer from the Consortium of European Life Sciences Research Infrastructures based in Austria. Michaela.
Hello, it's a great pleasure being here. I just want to double check that you see my first slide and that you hear me loud and clear.
Yes, but we can't see you.
Perfect, that's the plan.
All right. Fine. Okay, yep, we can hear and see the slides, yeah.
With this, I want to dive immediately in the presentation and of course share with all of you that the only sport we are likely all doing is conference hopping and unmuting and muting ourselves in presentations. And it's good that as life science research infrastructures that I'm here able to present, I can talk about more than muting and unmuting, but kind of the meat to the bone. I myself come from the research infrastructure BBMRI, that is the research infrastructure for biobanks and biomolecular resources. And I'm here to talk about the consortium of 13 research infrastructures in Europe. These research infrastructures have the mission to support cutting-edge science by offering access to their resources and services to European scientists from academia and industry predominantly. And the resources and services can simply by consultation, access to experts, but also access to data and biological samples, as well as use of data analytic tools, access to facilities, and support by technician and much more. What is exciting about that is that it kind of talks about what is mentioned in the previous presentations, that there is a lot of networking going on, there is a lot of research infrastructuring going on, and the aspiration at the end of the day is certainly global, but the reality of funding is a focus on Europe. And I want to stress with this slide that when it says here to European scientists from academia and industry, that is what we can guarantee. But wherever there is the synergy that is possible, either within a specific project or because of the relationships formed in other research endeavors, it is always used and made possible. And I will later use or showcase an example where it is done. So some of these services are even free for institution locations in the RI member states. And this, by and large, has been facilitated by member state contributions. So all life sciences that I represent here are funded by member state contributions and based in Europe with a European orientation. When we look at the map and the various sciences it represents, you see that it reaches from imaging, bioimaging to biobanks, to data infrastructures, to clinical trials and so on, and spans really across Europe. So not every research infrastructure has a representation in each and every member country, but as a group, we clearly can confidently say that we represent Europe. The main aspect of the activities that we do that also came out once again through a research project, namely Corbel, is providing access and continuing to provide access. So bringing the life science research infrastructures together to simplify the access pipeline, and that has some continuation in also one of the key follow-up projects, EOSC Life. And I do not have the time to show you the video in this presentation, but I will share it later on in the chat and encourage you to have a brief look. It is definitely worth it because it gives you a beautiful overview of what access pipelines exist across the life science research RIs. The key to all of it is the catalog of services that all the individual life science research infrastructures bring to the table. It started within the Corbel projects to, so to say, put it together, and continues after the project has ended on a joint website that should simplify and simplifies for the user to find what is needed. Samples, data, databases, technologies and facilities, models and tools. And more important also, as we've heard in the previous talks, training courses, expertise and support. At the end of the day, and here I cannot agree more, it is people and relationships that make it happen. Synergies are possible. Synergies can be pushed by the funders to some extent, but to keep them alive beyond the lifespan of a research project is due to the relationships formed. And in that sense, we can focus a little bit deeper on what services there are. And I would like to talk about the research infrastructure that I obviously know best. BBMRI services include IT services, quality management, LC, education and training. And the format that you see here on the page is the format that is true for every research infrastructure in the Life Science Research Infrastructure Consortium. So it should and helps the user to find easy access. Whom do I ask to get what I want and to satisfy the scientific needs at least. An example of how to accelerate research thanks to the life science research infrastructures provides yet again COVID-19. Again, a small video, which link I will share in the chat after my presentation. And it showcases how the various research infrastructures and their specific strengths working together, simplifying it for the researchers to whom to turn to in the end to get what they want, allows and accelerates research. In addition, individual RIs responded to COVID-19, and again, the website provides an overview of what the individual research infrastructures could do, from publications and modeling to services for COVID-19 vaccine developers to priority access to structure biology services for COVID-19 research. Out of 13 research infrastructures, it is always hard to pick something out, and therefore I deliberately want to draw your attention to the website to learn more. Again, it seemed fair to pick an example that I know from BBMRI, where we could relatively fast develop a COVID-19 filter in our directory. The directory contains sample and data resources from more than 600 biobanks and collection across Europe. But for rare diseases as well as COVID-19 collections, we had thankfully the mandate from our funders to introduce them and link them globally. So that is a nice use case how a pandemic like COVID-19, SARS-CoV-19, could lead to something that was at the start Europe-centric, and then out of necessity, because disease does not know any borders, developed further. And similar examples come from all of the other life science research infrastructures. More on the point for the life science research infrastructures, we aim to illustrate with success stories where joint support was able to help scientists in the interdisciplinary research. And the interdisciplinarity is maybe a keyword that I would like to stress more, in addition to the global aspects of what we face and the aspects of training needs, and at the end of the day, that to build and accelerate on synergies, it is down to the people and the relationship formed. More success stories you find on the website of Life Sciences RI. And the last item I want to draw to, because I'm a huge believer in collaboration as well as ample time for discussion in the plenary, is another initiative that came out from the LSRI activities, Corbel, EOSC Life, multiple projects, that is the European Alliance of Medical Research Infrastructures by BBMRI, RI-VIS and McEachern, where here some overlaps and natural synergies can be found as we collaborate on translational research, clinical trials, and biobanking, and the data and sample management, as well as the facilitation of data and sample research, especially in a clinical setting, is their key. This is already my last slide to provide you with the contact, whom to contact for kind of key questions on the LSRI community, including the website. And you see one of the pictures that I hope we can eventually return to, because as much as COVID-19 made us realize that, yes, we can collaborate in a virtual space, eventually it is needed to meet up again and form more relationships, to deepen them. And we always see what workshops, joint workshops can do across any scientific and any activity there is, as long as it is focused and we share the goal. And eventually we will meet, we'll meet again. That's my last word in terms of the presentation. Thank you.
Thank you very much, Michaela. So we do have a minute or two if anybody has a question now for Michaela. I noticed a lot of information and links have been put in the chat box. So I'm not sure, Natalie or Viri, if this information is available after the session that people can go back to. We don't want people disappearing onto websites at this time.
We will make it available.
We can put it on one of the community boards. So we'd encourage you to go and check out the Hoover community boards, and we'll copy some of the links from the chat onto there as well. Thank you.
Excellent. Okay. Thank you for that. I don't see any hands up or quick look in the box, any specific questions. So we might move on to the final presentation in this session from Stephen McEachern, who's the Director and Manager of the Australian Data Archive at the Australian National University. So I can see you there. Excellent.
Mm-hmm. Thanks, Stella. Yes, I'll get moving, and I'm just going to share screen now, so we'll see how this goes. Okay, So hang on, I'll just switch formats here. So I've got presentation mode running. Alrighty. Okay.
That's good.
Okay, there we go. Okay,
Yep.
thanks everyone. So I'm Dr. Stephen McEachern from Australian Data Archive. I'm based today in U. I'll just briefly start with a welcome country. I'm joining you from the lands of the Ngunnawal people here in Canberra, Australia. And I'd like to acknowledge and celebrate the first Australians on whose traditional lands we meet and pay my respects to elders past, present and emerging. Okay, so I'm not a life scientist, I'm a social scientist. And I'm going to talk a bit about some of the social science infrastructures and try and draw a link to some of the conversations that have already happened both yesterday, today and leading into tomorrow, and where social science fits within the frameworks here. It's also really looking at why it is the case that there might be some strong opportunities for both cross-domain collaborations on infrastructures, but also within some of the social science infrastructures in Europe and Australia as well. So a brief overview here of some of the history. What are some of the current infrastructures that exist in Australia and in Europe? Where some of that alignment exists? Where there might be some alignments with the social and the life sciences, and some possible future directions. Okay, so why is it the case that Australian social scientists are interested in Europe, and why are European social scientists interested in Australia? There is a long history of collaborative research between Australia and Europe in the social sciences, going back many years. Australia has significant cultural and social connections with Europe. We are, a colonised country out of the UK originally, but the population distribution of Australia actually is pretty strongly reflected in European migration as well. So about a third of Australia's population is born overseas. So of that 33%, about a quarter thereof, or about just under 8% of the Australian population as a whole. This was as of 2016. We don't have new figures just yet, but as of 2016, about a quarter of all residents born overseas were actually born in Europe, often coming in waves, particularly from the British Isles and Ireland early on, but increasingly from northern and then into southern Europe post-World War II. So there are over 1.8 million people in Australia who are actually born in Europe as of that last census, and that doesn't account for those who might have a parent or other born in Europe. So the cultural connections into Australia and the social connections, and as a result, a lot of the policy connections that occur between Australia and Europe are quite significant. Relative to, there's only about 60,000 born in the US and Canada, and a bit under around 250,000 each from China and India, coming from those countries. So it's a strong connection there. As we all know, Australia is part of Europe. I'd say I put this half jokingly, but it's not. In fact, I don't think it's coincidental that Australia ends up in the Eurovision Song Contest. But it's one of those reflections of actually that cultural connection that we actually have, and there are numerous of them, but it's the most most recent sort of representation of that. But I'd say the reflection there of that cultural engagement and the social mechanisms that exist are reflected in our participation there. The actual history of why Australia ends up in the Eurovision Song Contest is because we have a multicultural broadcaster here in Australia whose actions actually a licensee into the European Union broadcasting system. So we say again, we engage through the needs of our national communities. So that's also been reflected in a lot of the infrastructures that have actually developed in Australia in the social science and internationally. So for the European infrastructure folks on this session, Probably there are 3 European social science research infrastructures in the social sciences. CESDA, which is the Consortium for European Social Science Data Archives, it actually has a history back to its member organisations, dating back nearly 60 years. In the UK, in Germany, and Norway being the earliest of those. But CESDA has its origins actually in the mid 1970s. And Australia, actually, the organisation I run, the Australian Data Archive, was actually established in 1981. Leveraging on the approaches that have been developed around data sharing in Europe, and also in the what's called the Inter-University Consortium of Political and Social Research in the USA, to establish data sharing arrangements and agreements between countries. And there's a strong reason for this, is we actually want to study these comparative and cross-national cultural and social developments, understand how institutions and democratic practices might emerge between countries. And so in order to do so, we needed a mechanism for exchanging data sources between countries very early on in the piece. The sorts of data collections that go on. Certainly we rely quite a lot upon administrative data, but a strong tradition of conducting social surveys in particular, and as a means for capturing information from human populations on public opinion, attitudes and beliefs. These are the sorts of data, the collections that were occurring, and usually around relatively structured content, using tabular data resulting from those surveys. So there's very early on in the piece, we saw sort of the emergence of a fairly strong consensus around shared research interests, shared data requirements, and relatively shared structures as to how we go about analysing and using data. So that has resulted in a relatively close set of collaborations that occurred. They haven't been formalised in the way that some of the other collaborations we've been talking about here, but I'd say actually represent some pretty strong collaborations between countries in Australia and Europe over many years. So let's try again. There we go. So a number of countries actually established social science data archives across Europe and in Australia. It tended to be across a lot of the OECD countries, in fact. But they also tended to establish a set of related infrastructures at the same time. And so there's a relatively strong tradition of shared development of a set of related requirements. So there were data archives and repositories. There were often data collection facilities, usually for supporting national comparative and international comparative social surveys. And research methods training that went alongside that for the use of that data. And a number of the archives are often in institutions that offer summer training programs, national training initiatives, and these have led into some of these infrastructures as well. And then applied research centres, both for the use of data, but also for the development of new methodologies and techniques. And they've also been established alongside the international collaboration networks that allow us to align these cross-national activities. And so, as I said, CESDA, its original title was the Council of European Social Science Data Archives, and progressively, when the European ESFRIs and ERICs emerged, they were able to move in a relatively seamless way into coordinating to support one of the ERIC developments as it came through the European Union. There we go. And so, as I say, this is a kind of representation of that that emergence of these parallel sets of requirements that actually support collaborative infrastructures in relatively useful ways. So I'd say I've just taken a selection of institutions here. Some are European, but these also emerge in Australia, in Taiwan, in the United States, as a set of examples. There are a couple of similar ones in Africa and the like as well. Progressively over the past 30 years, and say we've established a fairly strong set of collaborative links between organisations we go through that could form the foundation for future collaboration into the future. So as I say, what we see is these four types of both infrastructure and the related training and user engagement services to allow us to leverage the infrastructure that was there going into the future. And then certain other services then start to emerge within particular countries or particular institutions, depending upon national practices, and they've tended to then roll out into other international collaborations as well. So the Germans at GASIS have developed a fairly strong experience in developing international survey support. The University of Essex, where the UK Data Archive is based, has established secure data services, and I think you're going to hear more about the Population Health Research Network tomorrow. There's been some collaboration there on, what are the demands on secure data services. Let's say as these requirements developed, let's say those needs were often shared nationally and internationally as well. So I say, so where are the opportunities then for some of those collaborations that let's say there are some that already exist, and then some that are pretty obvious opportunities for building up collaboration both within the social science and then into some of the other areas. And I'll touch upon where I think the life sciences and the social sciences really start to come together as I go forward here as well. So for the three European infrastructures that social science infrastructures that exist, there actually is, for two of them, a direct parallel that already is in place. CESDA has a parallel with my organisation, the Australian Data Archive. The European Social Survey, which is the national attitude surveys that occurs on a biannual basis. We have actually in fact run a coordinated survey with the ESS called the Australian Social, let's say what we call the Australian Social Survey International. And what that survey did was actually run a lot of those data collection had been occurring in face-to-face mode. We've been running international online web-based data collection, and that turned out to be particularly beneficial for the ESS team as COVID hit. There's a long tradition of doing face-to-face door knocking for data collection. What they were able to learn out of the Australian survey was, how some of the methods and how some of the instruments that they were using are transferred into an online environment as well. So that was both collaboration around data collection and around methodological development as well. And again, so Australia doesn't have a history of doing face-to-face, door knocking surveys because we're too big. The geographic, limitations work against that. We don't have a parallel of the survey of health and retirement in Europe. I'd say there has been some attempts amongst demographers and health researchers here in Australia, and an interest in the health and retirement study here in Australia. There are some related national collections, though, that might fit in pretty well into that. And say there would probably be some interest going forward in collaboration there. So there are some pretty clear integration points that have actually emerged over time as these common infrastructures have developed. One of them has been around shared data standards, the Data Documentation Initiative, which sits underneath CESDA and the ICPSR and Australia as well. It really came out of work around data sharing through the 70s and into the late 90s and was formalised in standard terms in 2001. There's been a lot of shared infrastructure because we have had common data formats and data standards. Dataverse is probably the best known of those at the moment. Previously, the Nestar system that was developed in Norway and the UK. There's shared content, similar styles of collections, quite diverse set of topics, but relatively standardized practices for methodology, research methods themselves, and also those shared research interests. So as I say, a lot of the methodologies that we use are transferred quite well across countries. So there's mechanisms, and we get quite a lot of cross-fertilization between Europe and Australia for these various reasons. And they find a nice basis for embedding things into the future. That said, Australian infrastructures do exist, but they're not embedded in national infrastructure, at least not yet. And you'll have heard a little bit about this yesterday from Simon Musgrave in the talking about the language data commons. I'm the lead on another of the projects within what's been titled the Hassan Indigenous Research Data Commons, which is a pilot investment from the Department of Education, Skills and Employment, to really establish a series of 4 projects. And quite literally, those project plans are under a review as we speak. I've got to provide feedback on one of those by tomorrow night. So this is about establishing a set of common packages, and there are some pretty clear parallels with European infrastructures here, with the Clarity infrastructure on the language data commons, and with the integrated research infrastructure for social sciences that I'm running, with basically the 3 different European infrastructures. We kind of fit in with a number of the developments in each of those infrastructures. And so I'd point you to Simon Musgrave to talk about the language data commons. So the work packages that we're developing really are designed to align with what is a pretty common research process in the social sciences, and it's a pretty common research methodology, more generally. So we've really been established a set of work packages that are designed to leverage national and international requirements. We'll be pointing to some of the European vocabulary services for picking up common and shared metadata, some of the design practices that occur in Norway for our we called our survey project research environment. So where possible, we're able to point to and leverage infrastructures that already exist where possible, and then send things back in the other direction as well. So increasingly, how do we coordinate those programs will form part of our thinking. But at this point in time, as I say, we're just waiting to get those approved. So I can't talk any further about that. As I say, we're just waiting for it to go forward. There are common data there are 3 basic themes that, where we see common practices there. Major collections, national population surveys, and particularly longitudinal panel studies, a lot of which have, and this is my lead into the life sciences side, increasingly linked data sources going with them as well. So a lot of the linked data work is around connecting survey data to other content. Some of it's administrative collections. It might be medical records. It might be one of the popular ones at the moment is in immunisation registers, tax records and the like, but increasingly into tying these also into biometric and other data collections. So as an example, we have the longitudinal survey of Australian children, which has a number of parallels in European studies. I think there's a plan for a cross-national cohort study currently in development. That's a survey data collected from birth to about age 15 at the moment for the young cohort here, but progressively they're linking in biometric data from what was called the Child Health Checkpoint from the Murdoch Children's Research Institute. They're tied into things like the GA for GH Genomics Alliance. Administrative data collections, geographic data collections, tying in, the parallels to the Inspire frameworks that are in Europe as well. So the sorts of problems that we're dealing with in those data linkages are common as well. And then we're also leveraging common frameworks for how infrastructures are developed. So this
4 minutes, Stephen.
Thanks. Yep. The most prominent of those at the moment we have in front of our Parliament, the Data Availability and Transparency Bill. Now we have an Office of the National Data Commissioner sitting in our Department of Prime Minister and Cabinet. This is a framework for actually establishing data sharing agreements of federal government data. Now, the foundations of that particular work is actually in the 5 Safes framework, which came out of the UK, a collaboration between their Office of National Statistics and the UK Data Archive. That's progressively moved across a lot of the policy networks inside government, particularly through national statistical offices. It went to New Zealand, progressively to Australia, and there's also a program of work, what was called Data Without Boundaries, across parts of Europe as well, which picked up some of this work too. But that framework is a mechanism of looking at risk profiling and for different aspects of data sharing, of sensitive data in particular. So now that we're looking with the Australian Research Data Commons, ARNet, who are collaborators here, and other partners on infrastructure to support that implementation, and we'll be feeding that back into the international environment as well. That project is CADRE, Coordinate Access for Research Data and Environments, and that's currently about halfway through our development as well. But again, these common frameworks, common problems, common interests are the themes that actually set us up for collaboration into the future as well. So lastly, there's my final substantive slide. Where is the opportunities for collaboration into the future? As I say, we could talk firstly on HAS specific infrastructure, and as I say, the most common of those is prominent of those is the HAS and Indigenous Research Data Commons. It has direct parallels with what's called SHOC in the European environment, the Social Sciences Humanities Open Commons, which is part of the EOSC program, and direct collaboration and integration with SHOC infrastructures would be a pretty clear basis for collaboration within the domains that we work within the social sciences. Secondly, within cross-domain infrastructure, there's pretty clear capacity. Researchers are pushing quite actively for working across the sciences. So the social sciences and the health sciences are pretty clear here, and Stuart touched upon a couple of examples of some of the population health interests there as well. But here we have also linkage and integration of common content, biometric, genomic sources, administrative sources, etc. And those sorts of things require common standards, and there's been some initiatives to try and establish that. There's a current program of work with the DDI Alliance, the Data Documentation Initiative I referred to earlier, and the CODATA, which is the data committee of the International Science Council, on establishing requirements for common standards. And then I think also on the underpinning infrastructure, the NRENs, PID infrastructure, cloud-oriented services more generally. I won't touch too much on HPC. I don't think that's a big demand just yet in the majority of the social sciences, but certainly what I would say is that is emerging, and that might be a direction as well. But certainly on the other underpinning infrastructures that are there, I think what we can expect to see as social science researchers start thinking more broadly about the sources available to them, particularly in social media, Internet of Things, these types of initiatives. That's when HPC also starts coming into the mix there, and where we might be interested in collaborative infrastructure there as well. But that sets a foundation for thinking about where some of the both within domain and cross domain infrastructure needs are looking into the future. And I'll leave it there.
Natalie, I can see you have a hand up.
Hey, I did post something in the chat earlier, but there've been a lot of link sharing going on since then. I was wondering about, from Stuart's presentation, that there's this initiative between the infrastructures to have joint appointments. And this is something that's come up in conversation in Europe, but we haven't, as far as I'm aware, we haven't done anything along these lines. I wasn't quite sure from your presentation if you've done it yet, or if this is an appointment that you've got open and you're looking for somebody for, and if you could share experiences on this and how it works and what the benefits are.
Certainly.
Thanks,
Well,
Stuart. Sorry, Stuart, just to say that I know Michael Dobby said he'd be happy to talk as well. So go first, and Michael, hopefully you're there. Yes, he's coming on. You can pin as well.
Sure. I'll go with a little bit of background as to where TIA's thinking is on this. And one thing we set up quite early and have now in place is a person or a chaperone to take care of each of our 3 facilities. So subject matter experts who've had some experience in the field or in a related field, who can talk the technical side to all 3 of our capabilities in a way that I can't, because my training isn't that broad, not remotely that broad. So, and from that kind of between the facility communication link worked really well. And it's something that I would really recommend to sort of have some sort of business development role and a industry broker role, and project attractant role. So there we have 3 of these roles, and their purpose is to go out into the world, find projects that need help and put them towards the right NCRIS, in the first instance, the right TIA facility that can help them. So that's where these BD roles have come from in our sense. Now, I think I'll just pass on to Michael in a second to say that we had a role who was half-time for various reasons. And what's arisen that Michael will go into more detail in is how he uses the other half of his time is in another, literally a joint role between two NCRIS projects. So I think Michael can describe a bit further.
Yeah, thanks, Stuart. It's a terrific question, Natalie. And to answer your question directly, we are exploring this. This person is already employed by TAA in a, I guess I think of it as an inter-facility role. So it's operating between the three arms to support TIA's activities. But what we are now stretching the idea to take, in this case, it's actually the same person, but it was intentionally looking for someone who could work between, so intra-facilities or between organisations with the same idea. It's been a long-held idea, but we've taken a while to actually come to explore how that could be organised. I think there's a lot to be gained, but there are obviously some risks in how you manage potentially competing priorities, how you manage time, manage co-supervision, if you like. But we're doing this in collaboration with BioPlatforms Australia. So you've heard a little bit about BioPlatforms. Our capabilities are unusually complementary. So I think this is a pretty safe bet. And within the month or so, we expect to have someone in post or at least being able to be contracted to start that position. It's really for, as Stuart said, to facilitate discovery or increase discovery or visibility of our facilities. So to have someone who's broadly across the range of capabilities that we offer, and to engage, effectively be a single point of contact to a single project or opportunity or mission, but to do that on behalf of multiple capability offerings, multiple, in our case, NCRIS research infrastructures. And so this would provide seamless access, but it also, I think, will provide a more timely access, but also more staged access to infrastructures. So you might start off with a BioPlatforms tool or laboratory, come across to Phenomics Australia, zip across to TIA and back to doing some preclinical testing with a mouse model or something like that through us. So there's a number of different ways that it could work, but I think it actually brings to life that schematic that Stuart has already presented in it today, to see how we can actually functionally work across our capabilities to have individuals who can basically unlock access in a seamless manner, in a coordinated manner.
Thanks, Michael. Were you following up, Stuart? Because Michaela...
Yeah, just very briefly, I'm following up on that and saying that it was said earlier on that it's infrastructures are people, and it's people that form the relationships. And I think having these kind of roles with these kind of skill sets in place builds those relationships, not just between the particular technical capabilities, but also between those capabilities and the broader community, and between researchers who may wish to access the particular capabilities. So it's really all about relationships. So the soft skills are coming to the fore again. I'll put my hand down now.
Thanks. Michaela?
Well, I can just piggyback on Stuart's comment, because I fully agree that this is indeed the relationships. And in order to be able to form those relationships, we've come to see what is possible in digital meetings, and eventually how much more is possible once we have met face to face. And one aspect that I would like to further highlight is the interdisciplinarity really in the field. I want to use this opportunity to out myself also as a trained social scientist working in the biobanking field since 2004. And it is exactly this interdisciplinarity aspect, that makes research infrastructures unique, and even more so the necessity, puts forward the necessity to have those translators, access managers, curators, and almost midwives in making it happen. And well, stopping my comment or concluding my comment with a personal anecdote is that I always had it relatively easy among the research infrastructures because I was always outside of my field, but no matter what language we spoke, all of us, we spoke science. And eventually, well, nobody could accuse me of asking a stupid question because I was anyways always the alien. So there is kind of a benefit in that. And I believe that those innovation managers and whatever multiple new roles will come into being from a competitive field, because as scientists, we are used to compete, but more and more in the research infrastructure field, in the service-oriented field, we have to collaborate and use synergies. And simply that we exist for more than 10, 15 years now is already a sign of that we do something right. Thank you.
Thank you. Thank you for that. Any of the other panelists like to make a comment on that? I guess Andrew and Niklas, do you have any shared personnel or thinking about that, or for your joint efforts?
I think it's a simple no, Stella. They would probably be very impractical given, I mean, the distributed nature of ELIXIR, the fact that all the scientific developments are done in our national nodes, and having a shared appointment between, say, Germany and Australia is probably rather quite complicated from tax logistics, but also from just general line management. So no, I don't think we've ever contemplated that.
No, I guess that would be right. It seems to me from the pictures that you showed, Niklas and Andrew, that you did have a lot of face-to-face interactions initially, and I agree totally with Michaela that they are so important to get things happening. And I think, Stuart, we found that with TIA when we had the initial facilities discussions, how the people who have been together more often came up with stronger proposals and ideas and things than the ones who kind of had just newly come together. Stuart.
Yeah, I think that's a function of the fact that they have it's trust. You know that you can raise an idea and know that it will be taken. It'll be addressed, it'll be looked at face on its merits without people, you know, wondering if someone has a hidden agenda or anything like that. So yeah, the people who have had that kind of set of relationships think more creatively and more in a more innovative fashion. Yeah, that we definitely saw that.
Yeah. Any other comments on that? I know the difficulties with COVID, and yet I don't think we would really be able to have this sort of meeting if we didn't use online virtual platforms. Michael's just put a comment in the chat box. Did you want to elaborate on that, Michael?
It's quite simple. I think I really quite like this as a premise on which to go forward, which is to really work hard at knowing as many people as we can, working hard to like those people, find common interests, and develop those trusting relationships and have that enormous portfolio to call on.
Yes, and I was going to point out that if you overlaid a map of Australia across Europe, it would cover it. So we operate in as large a geographical space as Europe does, with far less concentration of people and things. So the difficulties in communication often between the main centres are the same as probably you guys in Europe experience between countries. Stephen, what about in the social sciences and that sort of developing the collaborations and things? Is it all virtual, or are the people interactions important?
Oh, no, it's, yeah, the people side of it's definitely a challenge, and as I say, I mean, yes, I've been thinking about it a lot in terms of you know, what we can do as well. I tend towards an annual pilgrimage to so, you know, to Europe, that, you know, and figured on a, you know, 2 to 3 weeks every year, you know, knowing what how that worked. Interesting though, there might be some models where you can do some specific things. We actually, it was running a workshop last week. It was within Australia, but it was a week-long workshop, and it's so finding part of that in experience is sort of intense periods of times where you can have the opportunity to get to know people. Now, that did seem to work virtually, but again, the time zone considerations here are really going to work against us. So we did consider whether we could run, as I say, there actually was a parallel workshop that happened at the Dargstall Institute in southern Germany. We were looking at whether we could get some overlaps with that workshop, with the idea of having some parallels there. But the fundamentally the logistics worked against it in the end. It is, I think, within the same longitude, it's viable. It's how to deal with the time zone problems, I think, are really the challenge there. You can get the amount of time, I think it's right, but the at the same time is the hard part. So.
Thanks, Stephen. Niklas, you've got a hand up.
Yeah, I mean, it's also glass half full. So I think it is possible to overcome time zone differences. And actually, I think we've seen in the work that Andrew and his team has done with the Galaxy teams in Europe, for instance. And if you have people with strong common interest, where there is a direct benefit to collaborate globally, and there is it's a clear goal for that collaboration, so people know how they can engage and how to contribute, then time zones are a challenge, but they are not insurmountable. And actually that remote and collaborative work has been working amazingly well, Andrew. And I think it's that sort of clarity of purpose and the sort of very clear organization and the very sort of simple and clear mechanisms for engagement, so people know how they can slot in and contribute and be recognized for their contribution. And if you put those frameworks in place, then I think it is actually quite easy to do sort of work on a global scale, on the sort of 24-hour time zone difference of the planet. So I think, and perhaps that's where the learning, and that's where we need to invest for the future, is really to think about what are the structures, the frameworks, and the sort of people side of collaboration that help people to engage easily and sort of offset global travel and carbon dioxide with a bit more of planning and clarity, perhaps.
Yeah, I think that's a very good point, very good takeaway. We might change topics because we did cut off the discussion about voucher schemes and the way in which that can really help drive business to research infrastructure and supporting researchers. So I'll just get Stuart to maybe briefly go over TIA's voucher scheme, and then open up for discussion anybody else who has experience with voucher schemes and whether that's something that might work in cementing EU-Australia collaborations.
No worries, Stella. I think Vanna's question was first in terms of the fact that people have to bring some amount of funding. but considering it's only a relatively small amount in terms of 50,000 Australian dollars, you don't have to bring a great deal of funding. The other question before we moved on was how that works in terms of applications and numbers and things like that. What we're finding is that the number of applications we receive with every 6-month round, it's kind of self-limiting, because each applicant is a researcher from a company, a university, or a research institute. They have to have a conversation with the facility that they wish to access. So if nothing else, we've started a conversation just by virtue of there being an application process. And there is a kind of natural limit to how many projects a facility can handle. And we find that acts as a bit of an early filter, if you like, a bit of an intentional bottleneck that means only a manageable number of applications are received. And we know that each of the applications that we receive have talked to the facility they want to access. They've got some advice. They've started that way on the journey. So what that means is we end up with maybe our success rates vary, but it's generally about maybe 50–60% or so of applications are successful, which is way higher hit rate than a lot of competitive grants that are available in Australia. I don't know how it is in Europe. So we find it's kind of like a self-limiting feedback mechanism, such that you got a good chance of getting support if you can get to the application. And even if you don't, you've made contact with a facility that may be able to help you anyway. I should add one thing, that this is by no means the only way to access the facilities in the TIA consortium, but it's an additional subsidy on top of the subsidy that they should already be benefiting from through our funding. So it's a bit of extra on the top to help a project start where a project may not have started, but for a small amount of additional funding. And how we view the projects is that it's not like a grant where there has to be an outcome at the end, or a result or something like that. There has to have done something. The outcome for us is that the project has started. And we do ask for an outcome, what transpired from the project at the end. But the main metric for us is that we've helped a project start. And the more projects we can help start, the more likely we are the more likely we are to see projects that are progressing further down the development pipeline, hopefully to some sort of commercial or otherwise success. So we're finding it works pretty well, about 81 or so projects thus far. And we end up with off the top of my head, maybe a dozen to maybe 18 awards each round, something along those lines. That sounds about right. And yeah, they are open to overseas researchers and organizations as well.
Check out the TIA website. You might want to put that in the chat box, Stuart.
Oh yes, I will. I will do that if I can remember, if I can type out.
Any other panelists or people on this webinar have experience with this sort of system? No. Well, there has been, as I suggested, there has been quite a bit of interest in Australia about it, and in fact, TIA is partnering with another entity to provide help in the commercial space. Stuart, that's correct, the MRF.
Oh yes, that's something we're doing with a group called MTP Connect. They are in what's called an industry growth centre, which is an organisation funded by our Department of Industry to basically grow the sector using a number of initiatives, mostly funded projects or training and such like, but it's very much focused on the commercial realm. We're working with them to provide access to NCRIS infrastructure, not just ourselves, but also phenomics, bioplatforms, fabrication, other infrastructure. We are kind of providing, we're the infrastructure partner for them, and are able to sort of make introductions and organize some funding. It's early days and it won't actually be active for a year or so, but it's another way in which we're leveraging this scheme, which is not exactly, it's not our idea. There was a very successful state-based voucher scheme a couple of years ago. We're finding it works very well for us, but just to note that this initiative that Stella is referring to, it's a couple of years away, but it's a way of using our voucher scheme to provide access to a much broader range of NCRIS facilities, not just TIA.
Thanks, Stuart. I see Heather, who is one of our curators, cell and gene therapy, has got a question for you, Michaela. So maybe you could answer that about whether the use of facilities to member states is free of charge. That was Heather's question. And then I noticed you have your hand up, so you'd like to say something else as well.
Thank you, Stella. Yes, in essence, the services are free of charge for member states, and most of the ERICs have, and research infrastructures have, the model of members and servers. And whereas the services are by and large free for the member states, they are on the European translational level not free necessarily for the observers, because eventually we want all the observers also become members. And then at the end of the day, all research infrastructures are thinking hard on how to keep up the services that should remain open to the members, whereas others on the respective sites, and most of the research infrastructures are distributed ones, or have a mixed model between centralized services and distributed services, require for on-sites that have certain access procedures, but typically the services to the researchers are free. And I believe that Natalie wants to add immediately to my comment. So I would give the floor to her before I make my other comment.
Yeah, just very quickly, our own infrastructure, Instruct-ERIC, to member countries provides funding to access the facilities, the electron microscopes. So for the scientists, it's free at the point of access. It goes directly to support the facilities that are providing that access. And that's one of the benefits of membership of Instruct-ERIC. We are open to everybody in the whole world to apply, but that funding is only available to our members, or in a more limited restricted calls to our international collaborators with whom we've had MOUs signed. So we have an international call open at the moment, which would provide access on a similar basis to our international partners. And if anybody's interested in talking about that, my colleagues spoke earlier about getting in touch, but I'm very happy to hear from you.
Thank you, Natalie. Back to you, Michaela, for the hand-raising question.
Thank you. I cannot resist to make the comment that, Natalie, you look like from being from another world, which is mostly true in almost all the collaborations that we have, because they're always so efficient. But that aside, every access procedures may vary from research infrastructure to research infrastructure. However, all are committed to provide as much as possible with the given resources for free, especially noting that researchers do not get typically funding from their home institutions to access a facility yet, and depend largely on research collaborations. That said, a lot of brain power is going into the various research infrastructures, and whenever there is a joint call, as Natalie so eloquently put it, that is kind of the opportunity to provide not the full costs of access, but similarly to the voucher system that was mentioned, a benefit in facilitating the access. And ultimately, that's the shared goal, that the services that are out there are really used by the communities, and that's what ultimately the funders are also giving the resources for. From the BBMRI side, we aim in some instances to be as open as possible to the community, but it boils down again to the resources. And when it comes to the comment, it is really that the various models about voucher systems and collaborations are the aspect of the future, and collaboration also with industry. We're fairly much used to collaborate, us as research infrastructures, no matter where we are placed in the world, with our academic colleagues. We're also used to compete, but only a few of us have already good relationship with the industry and are used to the service character that the research infrastructures should provide. That said, I think we're on the right track, but I also think that we can do much more and learn from each other here. Thank you.
Thanks, Michaela. And certainly, I think when you're working with industry, there are quality standards and things that are required that are often not required in academic or not used in academic settings, which can actually cost quite a lot to have, you know, those sorts of standards reviewed and accepted and everything. So that's something that we always have to consider here. And I know many of our facilities here in Australia are accredited through NATA and other services, but it's a very important thing, I think. Michaela, were you responding to that?
Yes, first, fully agreeing with you, Stella, that the quality standards, whatever level they boil down to, as well as accreditation certifications nationally, internationally, regionally, become more and more important, are more and more required from industry. And that reminded me of one keyword that I also wanted to get across, that is the cultural change that we still live through as research infrastructures, as we're building up, that we provide indeed services to the community, and of course are facilitating science. But the run of the research structure cannot be done as a side project with a lot of blood, sweat, and tears. So we are in a phase, I believe, where the infrastructuring becomes more and more solidified, and that requires also a change in our mindsets in what we're doing. Never forgetting science, but defining clear roles, and that well, is best exemplified what has already been mentioned earlier with data stewards, access managers, and the like. New roles are needed. Thank you.
Thank you. Niklas?
Just to pick up on that and connect what Natalie wrote in the chat, because I think, I mean, it is right, of course, that research infrastructures, we are there to support excellent science, but they're quite large investments, and so of course they should be sort of directed to important sort of scientific or societal challenges as well. So I think that's the challenge-based approach, I think, is right. I think the difficulty is that it risks being donut-based funding with a big hole in the middle, unless we also come up with effective methods to charge the fully loaded costs to these challenge-based projects. And I think that's perhaps a sort of task that we need to take on ourselves as infrastructures to make sure that we sort of proactively propose mechanisms where the fully loaded costs for these type of projects can be passed on to the grants, because there needs to be a sort of a complete view of the whole landscape, and that might trigger changes within the infrastructures and in the way that we are operating and we are costing. But I think we also need to have a debate with our funders about what are the suitable models for funding.
Thanks, Nicholas. Stuart, you want to respond to that?
Yes, just to note that something that is quite common across NCRIS facilities is the idea of a tiered funding structure or tiered pricing for access. For TAA specifically, where we're into the product development area, where there's some specific IP on the table that needs to be protected and developed and eventually have its value realized commercially. A lot of the services are very bespoke and have IP provisions attached to them. So we find ourselves, for the most part, unable to consider any kind of free access, but that's just us. We're just one NCRIS project of 22. But in terms of the tiered funding, what we don't oblige facilities to abide by this, but we have a tier 1, which is researcher, basically university public supported researchers, what you might call meritorious or research of excellence. That's our access is charged at the lowest possible rate, sometimes just cost recovery. Often our funding to a facility means that the facility does not need to charge staff costs to an external project, for example. The second tier is usually for SMEs or university startups or very small, like sometimes single product companies, where there is still some subsidy applied, but it is to a lesser degree than for purely public funded research. And tier 3 is essentially the full commercial rate charged to kind of very large organizations and companies that can well afford it in terms of big pharma. So that's a model that is not universal, not even universal within CAA, but it's quite a common and useful way of conceptually framing access.
Thanks, Stuart. Any other thoughts about that? There was an issue about how one charges for access to publicly funded research infrastructure, but certainly in Australia, there's a very strong push by the government to encourage the use of the research infrastructure by companies, small, medium and large companies. So, and as Stuart's described, there is the 3-tier funding model. Andrew. Oh, Stuart, did you want to follow up?
Just small follow up, and just in the case that certainly in TAA's case, the level of funding that we're able to invest in certain facilities does not cover full operational costs. So to an extent, we're contributing and subsidizing rather than fully funding the operations of a facility. So we're a contributing funding partner rather than a complete sort of existential source of funding.
Thanks, Stuart. Andrew, and then Stephen's got his hand up as well.
Thanks, Stella. So one of the things that the BioCommons has been struggling with is how we represent to a life science audience, which is very rapidly becoming a data science. What the back-end research infrastructure should be able to do for them, and you know, referring to Niklas's comments, what it costs. And we've really, I guess, matured in trying to understand how we relate to NCRIS facilities which provide compute, and what that means for the researchers in life sciences who often want it in a different format. So they need data-driven compute rather than, I guess, model-driven compute. And we haven't worked it out yet. We're doing a lot of work with commercial cloud and whatnot. I don't think very many people in Australia in life sciences at all have a good understanding of the costs of doing analysis and how that should be met and who should meet it. I think we've got the answer either. It's a really tricky problem. Life sciences essentially has been a sort of a lab-based science for a long, long time. And it hasn't been, the cost of analysis hasn't been a problem. Now it is. And producing data is easy in some sense, and analysing it is the hard bit, and it has costs associated with it. And that's one of the things that we are really struggling with. I know that ELIXIR is scratching their head about this as well. How do you describe the costs of what is now developing into a data-driven science to folk who have been raised as a data-producing science?
Thanks. Yes. Yeah, I can see that people would struggle with having to make that change. A bit like funding, funding absolutely everything in your lab, whereas once upon a time, things seemed to appear on shelves and you could just access them. Now everything is costed. Stephen.
Yeah, it's interesting to pick up further on. I mean, Andrew and I have had conversations about how do you deal with. We have to deal with things that in the scale of tens of thousands rather than of small numbers, and so the social sciences are kind of moving into the same sort of phase. They're not so much actually for. It's still the case that a lot of what we do can be done on a desktop. It's moving away from that. But actually what's pushing us that way is actually sensitive data. Fundamentally is that, even if I could put it on my desktop, I'm not allowed to, fundamentally. And I'm guessing Merrin might talk a little bit about this tomorrow with PHRN. So it's a number of organizations that say this is where the cost models are becoming challenging in our area, has been, how do you deal with the fact that people don't know how to cost these things? The cost models that are emerging, there's a differential fundamentally between what it costs to operate. This is my sense of it. I say I'm generally on the receiving end of this. I'm a data provider for the most part, so I'm not providing the analytical services. But there seems to be a difference between what it costs to actually operate the infrastructure, whether it's on, whichever service, whether you put it on cloud or you put it on your own bare metal or whatever, and the what the individual researcher can afford. So, and fundamentally there's an aggregation problem here. Is the costs, if they can be aggregated up in such a way, probably can be managed at the level of the institution. But when they're managed at the level of the individual, there's fundamentally a gap in a gap in those two things, and what we haven't found is a mechanism in all cases for be able to deal with that aggregation problem. And I'd say I've seen a number of organisations who are trying to deal with that problem. The second reason that becomes problematic, though, is and the reason I mentioned the scale of how many people. If we're dealing with several thousand researchers who all want to do this, there's an administrative cost that comes along with this as well that I also don't think a lot of the infrastructures are ready for. I mean, I have enough trouble dealing with it, and I don't have to be running a really complicated access procedure there. It's still a click a button, type of model. But once we get to that model where you've got to get people into the infrastructure itself so they can work remotely, the administrative burdens of that are bringing together two problems, I think, that will be interesting to try and deal with. As I say, I'm yet to get my head around it yet, but I can see it on our radar fundamentally. But it's scale and security at the same time, I think, are going to create some challenges.
Yep, thank you. Thanks for that. Very important. Certainly the data security issues, and in the life sciences, you know, patient confidentiality and anonymity, all those sorts of issues make data sharing at times extremely difficult. Michaela.
I want to enlarge a little bit on the scalability argument, and really thanks, Stephen, for putting that forward, because the scalability comes also on so many levels. One of them you have mentioned, and the other one is on, say, ethical and legal services, where you can only do it on a case-by-case basis. So the question is, how can you scale it other than increasing awareness, providing education and training, go where the people are and not trying to get them to you necessarily, or focusing more on getting where the researchers already are, because you cannot add it as an add-on. It has to fit in whatever is existing. And that also means, again, a change of culture, a change of practices. And in order to make that happen, awareness of the needs, the roadblocks, and ultimately what can be done and what is indeed scalable needs to happen. Obviously, among us as research infrastructures for whatever services we are providing, whether they are tools or expertise. And then the other aspect is really to raise awareness not only to the users but also to the funders, because sometimes the scalability or the awareness raising is not only happening at the kind of upper or platform network level, but also on-site regionally. So how to make that happen is a discussion yet to be had.
Thank you. Do you have your hand up a second time, Stephen?
Yeah, I just wanted to follow up. I'm going to do a plug for the other project that I'm doing, because so the Cadre project I mentioned briefly is really starting to try and deal with that problem fundamentally, which is actually how do you deal with that exchange of information as say the administrative processes grow? How are we going to look at the informatics to actually exchange that set of information effectively? And we've been looking at across the social sciences and the health sciences. We're working with the ERIC team at University of New South Wales, amongst others, and ARNet, who are on this call too, to look at actually what information do we know that do we already know that we can exchange effectively and ethically. So there's a whole nother sensitive data problem that sits alongside that, but that's part of what that project is actually about, is a metadata exchange, so we can share that style of information effectively to deal with that burden that's coming down the line.
Thank you. Meryn, did you want to come on and speak? I know you've put an answer in the chat box. Hi, Meryn.
Hi, everyone. I did put an answer in the chat box, but I probably, if I am to speak, I might pick up Stephen's point about the complexity of using sensitive data. And I think that's a challenge in Europe and in Australia and in Canada and the US as well. And some of the data we use is used with consent. And then I was going to say it's easy. It's not really easy, but it's perhaps easier to exchange data once there's informed consent. The data PHRN holds is held and managed and used lawfully and ethically without individual patient consent. So that's an additional challenge to us, but we have got structures and processes that enable us to do that, as I said, lawfully and ethically, and having access to that data adds an enormous amount of value. So I don't think there's a simple solution, and it's something we're certainly grappling with. I think groups around the world, infrastructures around the world are grappling with it, and often the devil's in the detail. And the legal environment in Australia in particular is quite complex. In Europe with the GDPR, the good thing about the GDPR is it's very clear and it can apply across Europe. So unfortunately, Australia doesn't have that sort of legislation, which makes a sort of a jigsaw puzzle of legislation. But having said that, it is possible to navigate it. And I think Stephen sort of right up in the thick of things, trying to find a pathway for his particular project. But the idea of the 5 safes is it's a structure that's been well received in Australia, and it seems to provide an acceptable framework. So the issues of having the right researchers who've had the right training, using the right infrastructure in the right environment for an approved project seems to work quite well for us, and that's certainly something we're using. I'm not sure to what extent Europe has to go through that same sort of process. I know 5 safes is used to some extent in Europe and the UK.
Thanks, Maren. That's terrific. Thank you. I wanted to just briefly, probably the last topic we might cover off on, because I was very interested in Michaela's talk about what's it called, the catalogue of services that you provide for your infrastructure, and Therapeutic Innovation Australia also provides a way in which potential users can see what facilities are available. And I just wondered in general about how valuable that is to allow people from anywhere to really find out what is available. And I might, because you did describe yours and showed some nice pictures, Michaela, first, I might just ask Stuart to describe the navigator that TIA uses, which can be found on the website. Stuart? No, he's frozen momentarily. That's what happens in Brisbane when you're an hour behind everybody now. Michaela, maybe again you could.
Happy.
While Stuart rejoins.
Happy to do so. So in essence, various research infrastructures have various access pipelines, and the first step is, of course, always the mapping exercise. What is it? What is there? What services do they provide? You have a tagging list almost, depending on what you need to find out which research infrastructures provide them, and then you can look at them. Most successful have been open call pipelines in the context of a research project, and we have one ongoing in the project of EOSC Life within a system that was developed by one of the research infrastructures area that allows then to follow within one system the multiple pipelines, so that the user who comes in is not pushed to the individual research structure. That happens always also in the back end. Due to the funding limitation, the services is limited, or this open call for especially for sensitive data is limited to consortium members due to the funding structure, but it provides a very, very good model in how then ultimately in the mid to longer term open those access pipelines to researchers from anywhere. And the more we can test it out in a research project setting, I think the more beneficial it will be to the users that we ultimately want to reach.
Thanks, Michaela. I can see you there, Stuart. Have you paid your electricity bill? Are you back?
I stopped pedaling for a second. Yeah, I'm afraid I missed much of Michaela's answer, but I'm very interested in how that kind of directory and catalog works, because as Stella said, we have our own version, which we call the Pipeline Navigator, which is very much focused on life science and therapeutic development research infrastructure within Australia, and how it is articulated is as a pipeline, all the way from discovery, all the way through from discovery to lead optimization to preclinical testing to phase 1, 2, 3, and to the market. So it's presented in a very specific way that perhaps isn't super applicable to lots of other research infrastructures, but it's highly applicable to us and what we'd like to put out into the world. And I can just, if I can beg the indulgence of the chair, I could just give a very brief, actually, no, I'll put the...
Natalie is actually, she's brilliant.
Oh, thank you, Natalie. Yeah, that's great.
in the chat box, so people can have a look. Yeah.
Yeah, but I'm also aware that I think there's something called ORIN. I haven't been keeping up with this. Perhaps my NCRIS colleagues can correct me or follow up. But there is an emerging kind of catalogue that we're being asked to contribute to. It's still in its formative stages, and it's not ready yet. But it's there. I can't quite remember what it was called, ORIN or ARIN.
Michael's just appeared on screen. He's going
Michael, help me out here.
Yeah, it's was actually leveraging an existing navigator that's been designed for the Victorian region of Australia, based at Monash University, the Australian Research Infrastructure Network, but it's Victorian based at the moment. But they are starting to upload the National Research Infrastructure NCRIS facilities onto the database as a prelude to exposing them publicly or giving them greater visibility through that portal.
But that's
Thanks,
across
Michael.
the whole of the NCRIS network, all the way through remote sensing and marine biology and therapeutics and everything.
Yes. So yes, it will be interesting. Maybe as you can see behind me, I do have a therapeutic innovation logo. I'm the scientific advisor to TIA. It'd be really interesting, Michaela, to talk about the two ways of finding infrastructure, and if other people also have that sort of directory, because certainly we found that people were often unaware of what was available and certainly had no idea of how to connect to it if they did know. So these sorts of things I think are really important. Right, so Merrin says that RI-VIS is, that ARIN is in the exhibition hall, so we might all have to go there afterwards. Well, having mentioned that, normally at this point I would be saying there's just a couple of minutes of me between of you listening to me and going out for a drinks break, but sadly that's not the case now because we can't all meet afterwards. I'd just like to thank everybody. I think we've had a terrific discussion about life science infrastructure across Europe and Australia. Terrific to hear some of the collaborations that exist already, and hopefully we've made some connections that will allow us to develop more linkages over time, even allowing for the fine difference difficulties. So it just remains for me to thank all the speakers in the session and for everybody for posing questions and being engaged in the discussion this morning, tonight. I think it's gone really well, and I'll hand over to Viri to wind everything up.